Today in this session, I am going to talk about methylene blue reduction test. It is also known as MBRT. This test is generally used to test the quality of milk. It is a more common test or one of the most common tests used by the dairy industry like Mother Dairy, Amul, or by the food microbiologist to test the quality of milk. This session we will see that why milk is important, what are the different components of milk, what are the bacteria present in milk, how this bacteria can be controlled, and what are the tests which are done to study the quality of milk. It will help us to know about milk, its component, bacteria present in it, and what are the methods which we should follow in our laboratory to test the quality of milk. Milk and its derivatives are used every day in our lives for the preparation of different kinds of food. We know about these kind of milk products like butter, cheese, cream, and so on. Now these kind of milk products are not only highly palatable but they are also nutritious in nature. Now because not only the milk is an important nutrient for our diet or of our diet, but sometimes the bacteria present in milk can serve as the carrier of pathogen or as the source of pathogen in us. Milk is the fluid which is secreted by the mammals to develop and nourish their young ones. A number of animals has been developed whose milk can be used for human consumption. As for by now, cow is the most important animal in terms of commercial production of milk. As we can see, the main principal component of milk is water. Then comes fat, which comprises of about 4.4%, followed by carbohydrate. The main principal component of carbohydrate is lactose, followed by proteins. The main protein of milk is casein and whey. Due to the abundant presence of water, moderate pH of 6.4 to 6.66, and the presence of ample amounts of nutrients, milk is an excellent medium for the growth of number of bacteria. The bacteria can enter the milk during its different stages of handling of the milk sample.
What are the antimicrobial compounds that are generally found in milk?
The antimicrobial compounds generally found in milk are of different types. Apart from this, milk also contains immunoglobulins, popularly known as antibodies, and it contains lactoferrin as well as lysozyme. All this contributes to the antimicrobial nature of milk. The milk can become contaminated by different bacteria at the different stages of handling. It can become contaminated while it is present in udder, or while it is coming out from the teeth to its immediate surroundings, during its storage in uncleaned containers, or during the food handlers. That is when the milk is being handled by the different people at different stages of processing. The most commonly isolated microorganisms found in raw milk are of Streptococcus type, Micrococcus type, Corynebacterium, and so on. Let us now see how the bacteria can enter milk. So what are the sources? The first one, when the milk comes out from the teeth or from the udder through the teeth to its immediate surroundings. The teeth generally consist of its normal microflora. The microflora, when present on the pores of the teeth, can enter through the teeth into the udder and thereby contaminating the milk. The second one is there are a number of bacteria like Mycobacterium bovis, Escherichia coli, Listeria monocytogenes, which cause mastitis in cow. Mastitis is an inflammatory disease of the mammary glands, and as a result, the infected animals has this bacteria. Q fever, which is caused by Coxiella burnetii. Coxiella burnetii not only affects cows or other animals, it also affects humans. So again, similarly, if milk is ingested from a cow having Q fever, then the milk will contain Coxiella, and this Coxiella on ingestion by human beings will cause an infection in humans. So this is again another chance of the milk getting contaminated, and thus one needs to maintain good hygienic status of the milking animal. The other sources of contamination are from soil and water. Soil generally contains plenty of bacteria. The most common bacteria are Clostridium and Pseudomonas, which are residing in soil. The cow which is feeding on this kind of soil will ingest the soil containing the bacteria, and thereby the bacteria can have a chance to enter the milk stored in the udder. The water generally contains or might contain some enteric organism. Enteric organism like Staphylococcus aureus. So if it is present in contaminated water, the water will be taken in by cow, and this again bacteria will enter the milk. So in this way, the bacteria can enter the milk through another route, that is through the water route. Another important source is the food handlers. We, the human beings, generally contain different kind of microbes present on our skin. Most popular of which is Staphylococcus. Staphylococcus is present on the hands, fingers, upper respiratory tract. So during the different stages of handling, if Staphylococcus aureus is there present on the skin, so it might again contaminate the milk. Another important feature is hand milking. Hand milking allows the microbes which are present on the cow to be introduced into the milk. As for example, during hand milking, Escherichia coli might enter milk and produce a fecal odor or a fecal color. Again, one of the most common bacteria prevalent during the summer months is alkali genes viscolactis. Alkali genes viscolactis introduces a viscous climb to the milk. As in the case of some of the developed countries, as we can see in this slide, the hand milking has now been replaced by modernized, mechanized milking or milk handling equipment. As a result, there is minimum chance of contamination of the raw milk collected from the milking cow. As a result, it also reduces the total microbial load present in the milk and makes the milk a better quality to be taken in by the individuals or by the human beings. The raw milk contains different bacteria like Streptococcus, Micrococcus, Corynebacterium and so on. Apart from this, milk might also contain Staphylococcus epidermis and sometimes Flavobacterium. Now all these different kind of bacteria which are present in milk can be divided into non-pathogenic or pathogenic in nature. The non-pathogenic bacteria generally do not cause any harm, but their total population count should be reduced. Whereas the pathogenic bacteria, as we already know, are highly dangerous for any living organism, be it us also. These are the different sources of pathogen which can come or be introduced into milk. One is the animal itself, humans, or from the environment. So from the animal itself, we have Mycobacterium bovis, Brucella, Coxiella burnetii, Campylobacter. Now can you tell me what will happen when Mycobacterium bovis is introduced into the milk and that infected milk is ingested by us? Will it cause any harm to human beings?
It will cause infection.
It will cause infection. Here in this slide, we can only see two distinct types of milk spoilage. One is the curdling of milk and ropiness of milk. Often we find that the milk pouches which we buy from the market, if they are few days old, then sometimes on heating these old milk pouches, there is curdling of milk. The curdling of milk takes place because the raw milk or even the milk after pasteurization contains certain bacteria. bacteria like Lactobacillus lactis grew rapidly in this kind of milk. They take up the lactose present in the milk and produce acid. This acid precipitates the milk protein, that is the casein, and hence there is a curdling of milk. Another common spoilage is ropiness of milk. The ropiness of milk takes place because there is a change in the physical nature of milk. This kind of change is produced by capsulated bacteria like Klebsiella, alkali genes, and so on. This kind of bacteria deposit their gummy material into the milk, and as a result, the milk becomes sticky in nature. So these are the two most common spoilage which I have shown here. Apart from this, there can be plenty other or many other types of milk spoilages depending upon which type of bacteria is growing in the milk. Pasteurization, as the name suggests, was first developed by Louis Pasteur. So from there the name comes in pasteurization. Here in this process, milk after it is collected from cow is heated at a specified temperature for a specified period of time and immediately cooled to 4 degrees centigrade. This kind of pasteurization process not only improves the quality of food product but also increases the shelf life and the taste as well as the quality again of the food items.
Ma'am, does ultra-high temperature pasteurization have any adverse effect on milk?
Ultra-high pasteurization as such does not have any adverse effect on milk because in this process milk is heated at a high temperature of around 134 to 135 degrees centigrade for 3 to 4 seconds. As a result of this high temperature, there are two effects produced in milk. The first, the spores of Mycobacterium or any other pathogenic microbes are killed when subjected to this high temperature, as well as the total count of non-pathogenic bacteria is also reduced. The major disadvantage of this ultra-high temperature pasteurization is the quality or the nutritional status of the food product might be decreased. Because when milk is heated at such a high temperature, the nutritional components like the proteins might get denatured and there might be an altered taste of the milk or its products. Thus, ultra-high temperature is nowadays used by some dairy industries. Now let us come to what are the different methods which we can use for testing the quality of milk. There are three main methods: the viable count method, dye reduction test, and the direct microscopic count. In this method, the milk sample is serially diluted into test tubes. From this serial dilution or the different serial dilution, 1 ml of the sample is taken, plated on this culture medium. Following incubation at 37 degrees centigrade, the CFU colony forming units are formed on the plate and then the count is made, which indicates the total microbial load of the milk sample. The second is the dye reduction test. The dye reduction test is used when a redox indicated dye like methylene blue or resazurine is added to the milk. As a result, due to the presence of bacteria in the milk, there will be a change in the color of the indicator. If the milk is highly reduced, then there will be a color change in the indicator. If the milk is of good quality and containing less number of bacteria, then there will be no change in the indicator. So in this way, it helps us to determine the quality of milk. The next is the direct microscopic count. In this, a small volume of milk sample is loaded on this hemocytometer, and then the number of bacteria cell or the bacterial cell are counted under the microscope, and it is expressed as the total number of bacterial cell per ml of milk. In this way again we can get the microbial load of the milk. Let us now come to the MBRT test. MBRT, which stands for methylene blue reduction test. It is one of the most simple, inexpensive and a popular method of testing the quality of milk. If you go to mother dairy industry, you'll find that MBRT is the most common method which they use for studying the quality of milk. Methylene blue is a redox indicator, or it is a sensitive to the oxidation reduction environment of the milk. Raw milk contains bacteria. This bacteria will actively proliferate. As a result, the dissolved oxygen in the milk will decrease, and hence it will produce a more anaerobic condition or a reduced environment in the milk. Methylene blue remains blue in the oxidized condition. When it becomes reduced, it becomes converted to this leuco compound, which is colorless in nature. As a result, we will see the speed of disappearance of methylene blue when it is added to the milk samples. The speed of disappearance of methylene blue is directly proportional to the total number of bacteria present in milk. Thus, the principle of methylene blue reduction test is, it is based on the oxidation reduction activities of bacteria. So, we must first need a methylene blue dye solution. Concentration is 1 milligram in 50 ml of water. Apart from this, for performing this test, we have taken 3 different milk samples, abbreviated as MNP. M stands for 2 weeks old milk sample, N stands for 2 days old milk sample, P stands for fresh milk. As a control, we know that we have to keep a control for every experiment. So in this experiment, we are taking boiled milk as a control. Apart from this, we require sterile test tubes, we require a test tube rack, we require a glass marking pen, a spirit lamp, 10 ml glass pipettes, 1 ml pipette, and a mechanical pipetting device. It is done that first we take the sterile test tubes, label them sequentially. Now all these kind of tests are generally performed in duplicates. That is, if we take the sample M, we are taking 2 duplicates or making duplicate sample of M. Similarly for N, similarly for P, and 2 for control. Using different 10 ml sterile pipettes, we are transferring the milk samples from the beaker to the sterile test tubes. Afterwards, which we are adding 1 ml of methylene blue to all the milk samples. We insert the stoppers and then mix the samples or the milk samples properly with the methylene blue. We keep this reaction mixture at 37 degrees centigrade and record the rate of disappearance or the speed of disappearance of methylene blue after every 30 minutes interval till a specified period of 6 to 8 hours. Thus, in this way, we can test whether the milk sample is able to discharge or is able to decolorize the methylene blue. We record the observation in this kind of a chart. Here the time in hours is mentioned, here the milk samples. Since we are taking 2 milk samples or duplicates of each milk sample, so there are 2 columns of M. Similarly for N, similarly for P, and similarly for C. We find that during the different hours or time in hours, the discoloration takes place. First is the milk sample, which is abbreviated as 2 weeks old or as M. Discharge the methylene blue or the color of methylene blue takes time of about 1.5 hours to 2.5 hours to discharge this color to white. Similarly, for 2 days old, it takes time of about 2 hours to 3 hours in order to discharge the color of methylene blue to white. Similarly, for fresh milk, we can find that there is no decolorization even after a period of 6 hours. Why are we saying that we are taking boiled milk as control?
Because it does not have any microbial load.
Why it does not have any microbial load?
Boiled pasteurization has been done, or it's been heated to high temperature so that the bacteria are killed.
Yes. Since we are heating the milk, the microbes are killed. And as a result, there will be no microbial activities present in this boiled milk. As a result, methylene blue will remain as blue in color. So, accordingly, we can see the time which is used or which is noted down for MBRT equation or reaction. Now, how can we classify the milk quality as fair, good or bad? If the methylene blue reduction takes place immediately within 30 minutes, then the milk quality is of very poor type. If the reduction of methylene blue takes place between 30 minutes and 2 hours, then the milk quality is of poor type. When the reduction of methylene blue takes place between 2 hours and 6 hours, then it is of fair quality type. When the reduction of methylene blue takes place between 6 and 8 hours, then it is a good quality milk. So, depending on the rate of disappearance of methylene blue, we can judge the milk samples as good, fair or bad, as well as we can differentiate between the milk samples. So, thus what will happen? Since the M sample discharges the color between 1.5 to 2 hours or 2.5 hours rather, so the milk quality is fair. The N sample discharges the methylene blue color between 2 to 3 hours, so the milk quality is again fair. Whereas the fresh milk does not decolorize methylene blue, the milk quality is of good type. So, in this way, we are able to differentiate the 3 different milk samples provided to us. The precautions which needs to be followed are light speeds up the reaction of methylene blue reduction. So, the test tubes should be covered. The second is the concentration of the dye should be uniform. The third is there should be no crimming of the milk samples as it removes the bacteria from the milk samples and causes an unequal distribution of bacteria in the milk. These are some of the precautions which needs to be followed during performing this MBRT test. Resazurin, again similar to methylene blue, is a redox indicator. Resazurin is in the oxidized form, it is slate blue in color. When it is slowly reduced, its color changes from slate blue to pink to colorless. Now, similarly, when we add resazurin to the milk samples, we can find a different array of colors. After it is being added to the milk sample, if it remains blue in color, it indicates good quality milk. If the color changes to pink or to light pink, then the milk quality is of fair type. If the milk becomes colorless after addition of resazurin, then the milk sample is bad. So, in this way, again, we can differentiate between the milk samples.
Ma'am, which test is better, methylene blue reduction test or resazurin test?
Till now, I have discussed about 2 dye reduction tests. One is methylene blue, another is resazurin. Methylene blue is the most common and popular method of testing the quality of milk. It is regularly practiced in the laboratory for testing the quality of milk and even by the dairy industry. So, thus methylene blue is much more popular than resazurin and much more accurate than resazurin test.
Other than resazurin test and methylene blue reduction test, are there any other methods to assess the quality of milk?
The quality of the milk apart from performing these two dye reduction tests, can also be determined by the use of litmus milk reaction. In litmus milk reaction, litmus dye is added and it helps us to differentiate between the organisms present in milk. That is, whether the organisms are producing acid or not. Because if it produces acid, the pH decreases and hence there will be a color change in the litmus reaction. So in this way it helps us to differentiate between the organisms present in milk. So thus the dye reduction test is a very important test. It helps us to differentiate between the quality as well as between the milk samples. The major advantage of the dye reduction test is mainly the viable cells, they show this kind of reaction. The greater disadvantage of this method is some of the food products cannot be subjected to dye reduction test because these kind of food products contains reductase in them. And as a result, there is every possibility that the methylene blue test will give a false result or will be a false positive. Milk is an important constituent of our diet. Milk, due to the different stages of handling, can become contaminated by different kinds of bacteria. The kinds of bacteria present in milk can be of non-pathogenic or pathogenic types. History has witnessed a number of milk-borne pathogens which have caused catastrophic disturbances in the human lives. It has even claimed plenty of lives. In order to remove the pathogenic bacteria from milk, a common process known as pasteurization is generally performed. Apart from pasteurization, there are other methods to test the quality of milk, amongst which the dye reduction test like MBRT is the most common, popular, accurate, simple and inexpensive method to test the quality of milk.